In this video, I wanted to discuss the process of photosynthesis, where effectively you're looking at two sets of reactions. One of them are known as the light-dependent reactions, where light energy is being absorbed and is going to be used to charge up molecules that we saw from our process of glucose catabolism, our ATP and our electron carrier NAD. Those energy-rich molecules that are being generated from our light-dependent reactions are going to get used in a cycle reaction over here known as the Calvin cycle for what are known as light-independent reactions. This is where the process that we traditionally think of as photosynthesis is going to occur, but this is actually the synthesis part. So it really is two stages, the photo reactions and the synthesis reactions. And if we look at this process, it all starts with pigments that are buried in membrane complexes. And you might have heard of chlorophyll. This is a pigment that is found in the membrane of what are known as photosystems. And in the center of this photosystem is this chlorophyll molecule that actually will absorb the light energy that is out there. And that light energy is going to get transferred to electrons, and it's going to boost their energy. So you can think of this electron in what's known as the ground state. It's just sitting there. It doesn't have a lot of energy available. And when we shine light on the chlorophyll molecule, it's going to transfer that light energy to our electron, and instead of being in the ground state, it's going to be boosted. So now your electron is bouncing around kind of like a little ball that has absorbed a whole bunch of energy. And if you think about our process of glucose, those high energy electrons are being stored in glucose that is broken down for our catabolic reactions that we saw in the previous chapter. So if we look at this process, we need to keep generating these high energy electrons, so you have to replace those high energy electrons. And in this photosystem that is numbered number 2 because of the order of discovery, not in terms of how they're used, photosystem 2 is going to split water molecules, as we can see in this reaction down here, and that's going to be used to replace the electrons. So by splitting water, the opposite of what we saw with cellular respiration in the electron transport chain, where we dump the electrons onto oxygen and generated our water, here we're running the reverse reaction. The photosystem is going to split your water molecule, and that's going to generate our electrons that can be boosted in the photosystem right here. And the byproduct of this is our oxygen molecules that we need for those electron acceptors at the end of the process of our electron transport chains for cellular respiration. If we look at where do those electrons go, well, there's a whole complicated electron transport chain that comes out just like what we saw with our glucose catabolism. Over here we can see there's photosystem 2. It's boosting those high energy electrons, and they're going to go through an electron transport chain similar to what we saw before. The electrons are going to bounce from one molecule to another, and they lose energy through this process, and that energy is going to be used again to pump protons across the membrane. So very similar to what we saw with our glucose catabolic reactions. That proton gradient is going to build up more and more, and those protons can then travel through our ATP synthase molecule just like we saw before, and that's going to be used to generate our ATP molecules. So here, the first part of these light-dependent reactions, we're generating oxygen with our photosystem 2. When we split water, it's producing energy, or it's producing electrons that are going to get boosted with the light energy, and that's going to drive our proton gradient for ATP synthesis. And when we look at this process in cells, they're going to actually be able to use these electrons for the other photosystem known as photosystem 1. Same idea. Chlorophyll is going to absorb that light energy, and that's going to be used to boost our electrons a second time, and then they're going to get transferred onto our electron carrier. In this case, NADP. It has an extra phosphate added to it, and we're going to make this NADPH. So again, two energy-rich molecules are coming out of this process, either ATP or NADPH, that are going to be used in the dark reactions or the light-independent reactions. And if we look at our video, similar to what we saw before, you can see how this process, which is hard to see from those pictures, but you can see the stepwise fashion of this whole process. Photosystem 2 absorbs the light energy and boosts those water electrons for our electron transport chain that's going to drive our proton gradient being formed. There is your water behind the dam that we saw before. And when the water goes through the turbine in the dam, in this case ATP synthase is our turbine that spins, that's going to be used to generate our ATP molecules that are going to go into the other set, the synthesis reactions. And again, those electrons that came out of our first photosystem over here and the electron transport chain, they can get reboosted and go over here to this separate kind of electron transport chain where they're going to get dumped onto our other energy or our other high energy electron carrier molecule NADP to make our NADPH that also goes into that dark reaction or light independent reaction. So if we look at this process here, we're going to have those two parts of photosynthesis that are linked together. The photo part is your light energy being stored in ATP and NADPH. And where does this process take place? It takes place in the inner membrane of our chloroplast. Just like our reactions for glucose catabolism took place with the electron transport chain and ATP synthase, that was in the inner membrane of our mitochondria. Well, here, the inner membrane of our chloroplast is the thylakoid membrane. And these membranes will get formed into stacks that are known as grana. And then those molecules that are coming out of this process, all of those energy-rich molecules are going to go into our synthesis reaction over here that's known as the Calvin-Benson cycle. So again, you can see photosynthesis really is the light part with the photo and the synthesis part that takes place in the presence or absence of light. And this is a series of reactions where effectively the chloroplast is going to use the stored chemical energy that was available from our ATP and our NADPH that are produced in the light reactions, and they're going to pump that into a series of reactions where we're going to fix our carbon dioxide molecules together. And so if you look at this process and think about what's going on there, that carbon that we saw from when glucose was catabolized, it was oxidized. We stripped the energy out of those bonds between the carbon. Well, what is the synthesis doing is basically fixing those carbons. By using the energy from these molecules, we can start to glue our oxidized carbons back together, and we're going to be making these high energy bonds by transferring the high energy electrons from our ATP and our NADPH. And those are going to be used to run through this Calvin cycle, and the output that comes out of here is the sugar. And so if we look at this, the light reactions are taking place in these membranes right here. Again, our membrane sacs that are on the inside of our chloroplast. And then those high energy molecules are now floating around this fluid known as the stroma. Carbon dioxide comes in there, and the Calvin-Benson cycle is going to fix those carbons, and you're going to get your sugars. So this stroma around here, you can think of, we've got our pancakes for our grana stacks with the thylakoid membrane, and then you've got sugar around the outside. And it's just sort of a convenient way to remember this process and what's going on there. So think about, what does a bacteria do? They don't have chloroplasts, but if you look at our endosymbiotic theory, mitochondria and chloroplasts were once independent bacteria that were engulfed by a eukaryotic precursor cell. During this process, they picked up that outer membrane layer that we see in both mitochondria and chloroplasts. What used to be the bacteria cell membrane is now the inner membrane for these organelles in our eukaryotic cells. So the site where these reactions took place with prokaryotic cells is actually going to be that cell membrane around the outside. The light-dependent reactions are going to take place in the cell membrane, which folds up. And so it's very similar to the folds that we see with our thylakoid membrane. You're going to have pigments that are available to absorb that light energy and generate these energy-rich molecules that are going to float around in the cytoplasm for the bacteria cell there. And then you're going to be able to run those light independent reactions, the Calvin reactions, where you're going to have those carbon molecules getting fixed together. And if we look at the sum of all this, what we can see is that carbon is cycling constantly between the environment and between living organisms. And these are reciprocal reactions that are out there. We are consumers. We are heterotrophs that are going to eat our food and break it down. And if we look at this process, there's your glucose. With a little bit of oxygen, we're going to be able to strip the energy out of these glucose molecules and oxidize those carbons. And that energy is going to be used to recharge the cell's batteries. So we're animals. Also, if you look at things like fungi, like mushrooms and molds, and little single-celled animal-like cells that are known as protozoans are all going to use this process of cellular respiration. Oxygen gas is going into this process, and CO2 gas is coming out of this process. But remember, your oxygen is being used to generate water because it is an electron acceptor. On the reciprocal side of this process, we can see that there's a group of organisms known as producers. They are also known as autotrophs because they can automatically make their own food, and they will literally feed themselves. These include things like plants and algae and also cyanobacteria that can use photosynthesis. And if you look at this process, it's the reverse of what we saw for the heterotrophs over there. These organisms will take oxidized carbon and split water molecules to get electrons, and then light is being used to boost the energy of those electrons. And that energy is stored up in the carbon bonds of our glucose molecules over here. And the oxygen that is being produced that we need for cellular respiration is coming from the splitting of water, which is the opposite, or again, the reciprocal reaction for what we see when we look at this process of cellular respiration. So if you think about it, this constant cycle is going on with carbon. The carbons are storing up energy by the producers, the autotrophs, and then the consumers, or the heterotrophs, are getting the energy so that their cells can recharge their batteries and do this process. And the other important thing to remember is our producers out there that are eukaryotic cells like plants and algae, they are using the chloroplast to make that glucose and then using that to feed their mitochondria for the cells.